Now we're going to go ahead and start talking about Down syndrome. And another name for the condition of Down syndrome is trisomy 21, meaning that you have 3 copies of 21. Remember that word somy is talking about aneuploidy. Let's go ahead and get started. So before we dive into Down syndrome, I'm going to take a moment to talk about a Robertsonian translocation, since this is really important in familial Down syndrome. So Robertsonian translocations happen with acrocentric chromosomes. And an acrocentric chromosome looks like this. It's whenever you have a long stick, the centromere, and just a tiny little bit sticking out at the end. And if you have 2 of those, and keep in mind that these would be non-homologous pairs. So at translocations, we're talking about non-homologous pairs. That means I'm talking about like chromosome 1 and chromosome 7, or in the cases of Down syndrome, chromosome 14 and chromosome 21. And the 2 long ends are going to attach together, and the 2 short ends are going to attach together. And those 2 short ones, when they attach together, they're going to be lost. And this is because they're so tiny, the cell doesn't really pick it up. So we know that we need like spindle fibers to attach to the centromeres and all of that stuff. And it's just really, really small that the spindle fibers can't find it. And so it ends up being lost and we don't have it anymore. So like I said, this is important in familial Down syndrome. So what does this look like? So we have our acrocentric chromosomes drawn out already. And if we had a Robertsonian translocation, we would have our long ends attached together and our short ends. So let's talk about a couple of things with this before we move on. First, think about how much information is located at the ends right here, like the small purple and the small blue. There's really not a lot there. Keep in mind that not all of your DNA is going to actually code for something, and a lot of it is just repetitive sequences and like that kind of stuff. There's also those telomeres at the end that are just protective. So that little bit of information actually isn't that important. And there might be like 1 gene there, maybe 2 that are doing something, but for the most part, there's not really anything there that's that important. And the majority of your information that's important is in these long sticks. So even though we lose this little guy right here, because he's so small, we still have pretty much all the information that we need. We still have everything pretty much from blue and everything from purple right here. So I know there's a little bit lost, but that information just isn't that important. So we still have everything. So do we really lose any information? No. But if we were to count up the number of chromosomes that we had, so in a cell, if you were to look at these 2 right here, you would say that's 2 chromosomes. And this right here is just 1 chromosome. So if you're looking at a karyotype, you would say like, oh, there's 1 less chromosome. So this would be a monosomic individual, -1. And while they would still be monosomic because they are -1, they haven't lost any information. So that's a really important consideration between this and non-disjunction, where you're actually losing an entire chromosome. We haven't lost the chromosome, we just attached 2 together, so now it just looks like 1 chromosome, even though it's still 2 things of information. So hopefully that makes sense. Let's talk about Down syndrome now. So there's 2 types of Down syndrome, primary and familial. Most likely you guys are more familiar with the primary type and you not the familial. So first let's talk about how they're similar. Both are going to have 3 copies of chromosome 21. Because with Down syndrome, it's also called trisomy 21, we need to have 3 copies of chromosome 21. But then how do they differ? Well, they're going to differ on where those 3 copies are, so the location of those 3 copies. In primary, the one that you're most familiar with, all 3 are going to be separate chromosomes that are located at the 21 location. So if we had our karyotype at location 21, you would have 3 chromosomes right there. And because we're only supposed to have 2 per location, now we have 3. That means they're going to be trisomic, and they would have 47 chromosomes. Now, familial, you still have 3 copies of chromosome 21. but the location is a little bit different. Two of them are at the 21 location, just like normal. Which originally, if you're looking at that, you're like, okay, there's not a problem there. There's only 2 copies of 21 and I want 2 copies. The problem is that the third one is going to be attached to chromosome 14. So we would have a regular chromosome 14, and then you would have a chromosome 14 that had a chromosome 21 attached at the end. I'm going to make all these chromosome 21s purple to make it obvious. So there are still 3 copies of chromosome 21. The location is just different. So if we were to look at a karyotype of someone that had familial Down syndrome, you'll notice there's still just 2 chromosomes at every single location. And this means that they're going to be counted as having 46 chromosomes, because that third copy of 21 is just attached to an existing chromosome. So if you were to just count, you would still count 46 versus primary, where you have an actual extra one that's on its own. So there's something else that we talk about with familial Down syndrome, and that's called a carrier. So this individual would be a carrier for Robertsonian translocation, and they don't have Down syndrome. So remember, carriers aren't going to be affected by it, but they can pass it on to their children. So an individual that doesn't have Down syndrome, we know they have to have 2 copies of chromosome 21. And in a normal individual, usually those 2 copies would just be at the chromosome 21 location. In a carrier, they're going to be in a slightly different location. So at the spot 21, they would have 1 and only 1 there. And at the 14 location, they would have their 2 chromosome 14s, and their second 21 would be attached to that 14. So they still have 2 copies of chromosome 21. One of them is just attached to chromosome 14. So now think about what a karyotype for this individual would look like, and if we were to count everything up. If you were to count everything up, you would count 45 chromosomes here. So this individual would look like they were monosomic. And keep in mind that here, monosomic means -1 chromosome, but that chromosome isn't actually missing. It was just added onto another chromosome. So we didn't actually lose any information, and that's why this individual would appear phenotypically normal. Okay, so the next thing that we're going to do is draw what someone that is a carrier for Robertsonian translocation between 14 and 21 would look like whenever they mate with a normal individual. So I went ahead and pre-drew some of the circles here, just to make it a little bit easier. So if you are someone that likes to draw along or anything like that, go ahead and pause the video for a second and just make these circles look like this, since this is the chart that we will be following. So let's see, I'm going to make my chromosome 14 blue, and I'm going to start with my normal individual. So my normal, I'm going to label it. This is chromosome 14. My normal individual should have 2 chromosome 14s, and I'm going to make my chromosome 21 green. And they should also have 2 chromosome 21s. So a normal individual is a diploid. They have 2 of everything, 1 from mom, 1 from dad. Now, if that individual were to form a gamete or a haploid cell, we know that we go from 2N to N, and we just get 1 of everything. So we would get 1 of those blue sticks and 1 of the green sticks. So we would get 1 chromosome 14 and 1 chromosome 21. And that would be what a normal haploid would look like. And keep in mind, if this were to form with another normal haploid, with 1 blue and 1 green, they form, now there's 2 blue and 2 green, and that's normal. So now let's look at what a carrier for a Robertsonian translocation between chromosome 14 and 21 would look like. So they would have a regular chromosome 14. They'd have another chromosome 14, except this chromosome 14 has the Robertsonian translocation, and they would have one chromosome 21 by itself. So keep in mind this carrier looks the same as the normal person, phenotypically normal. Because if you look at the actual information that's there, it's the same. 2 copies of 14, check. 2 copies of 21, check. The difference is that in the carrier, a 14 and 21 are combined. So with our normal one, it was super easy to make the haploid because we literally just took the 4, we divided by 2, and we got 2. We send one blue to one cell, a blue to the other cell, a green to one cell, a green to the other cell. But whenever we have 3, it's a little bit different because you can't just split it evenly because there's 3 things. So whenever we split this, there's going to be some cells that end up with 2 and other cells that end up with 1. So we're going to draw what each of these scenarios could look like. So for example, we could have the green and the blue end up by themselves. So remember, we're splitting everything up into 2 and 1. So we can have those 2 go together, and that'd mean the cell that would be like the complement of that would have that third one, our Robertsonian translocation. So that's one option. Or we could split it up another way, and we could say that I'm going to have my chromosome 21 and my Robertsonian translocation go together. And I'm going to have my chromosome 14 by itself. Or I could say I'm going to have my chromosome 14 and my Robertsonian translocation go together. And I'm going to have my chromosome 21 by itself. So notice that each of them gets a chance to be by itself, and then the other 2 are combined. So these are some options of what the haploid cells could look like. So in order to form a child, we need like the egg and the sperm to combine, 2 haploids to combine. So what I'm going to do is I'm going to take this haploid and I'm going to combine it, the normal haploid, with each of these options and draw the zygote right here. That would include combining that haploid cell from the normal parent with each of these carrier cells, or the cells that came from the carrier. So looking at this first one on the very left, I have blue-green combining with green-blue. So I would have 2 blues and 2 greens, aka 2 chromosome 14s and 2 chromosome 21s. In my second one over, I'd have a blue, a green, and then a blue with a green, or I'd have that Robertsonian translocation. So I'd have blue, oops, that is the wrong color. Blue, and then I'd have my Robertsonian translocation, and my green. So the blue and the green came from the haploid normal parent, and that Robertsonian translocation came from the carrier. So then in my next one, I would have blue and green from the haploid, and then translocation, Robertsonian translocation from the carrier. Let me go to my laser pointer. So blue and green from the haploid with Robertsonian translocation and chromosome 21. So if I draw that out, I would have something that looks like that. And I'm going to keep going to the end. So in this next one, I would have a blue and a green combining with a blue. So I'd have 2 blues and 1 green. In the next one, I would have a blue, a green, a blue, and that Robertsonian translocation. Wrong color again. So I have those 2 blues, one from the haploid normal parent, one from the haploid carrier, and then the Robertsonian translocation. And the last one would be blue, green, green. So I'd have blue, green, green. Okay, so now let's look at the viability of these. So something to note first is that we need at least 2 copies of everything in order for these to survive. So this, like 4 chromosomes 14 and 21, you need at least 2 copies. With your sex chromosomes, like Turner syndrome, you don't always need 2 copies, and that has to do with X inactivation and that kind of stuff. For our autosomals, we're going to want 2 copies of everything for these. So that means that if I only have 1 copy of chromosome 14 or only 1 copy of chromosome 21, they're not going to be viable. So that accounts for this one and this one. Those are going to be non-viable. The other one that's non-viable is going to be the one right between those 2 X's because it has 3 copies of chromosome 14. So you might be thinking, okay, well, why is 3 copies of chromosome 21 okay, but 3 copies of chromosome 14 is not? That doesn't make sense. And the reason why is that if you were to look at a karyotype, you'd see that chromosome 21 is pretty small, be like that big, and chromosome 14 would be like that big. So chromosome 14 has a lot more information. So if we have this much extra information in our person, they're not going to make it. If we have this much extra information in our person, they're going to make it, but there's going to be some developmental issues there. So this last individual is also not going to be viable because that's just too much extra information. So let's look at the ones that are left. These are the ones that are viable. So looking at the one on the far left, what do you notice about it? There's 2 copies of chromosome 14 and 2 chromosome 21. And that's all there is. And there's nothing weird about it. They each are in their own separate location. And this looks just like our normal person. So this means that this one right here would be normal. The next one, let's see what we have. We have 2 copies of chromosome 14, and we have 2 copies of chromosome 21. But the weird thing here is that a chromosome 14 and chromosome 21 are attached. That's our Robertsonian translocation. So this one person is still going to be normal, but they are going to be a carrier. So they have the same amount of gene information as our zygote on the very left side, but they have those 2 attached. And then we have our last individual. You have a chromosome 14 with 21 attached, and 2 21s. So how many copies of chromosome 14 do we have? 2. How many copies of chromosome 21 do we have? 3. 3 copies of chromosome 21. That means this is a type of Down syndrome. Let's figure out which one it is. If it's primary Down syndrome, that means that these 3 copies are individual by themselves at chromosome 21 location. In familial, you have 2 at the chromosome 21 location, and the last one is attached to chromosome 14. So what's happening here? This one is going to be familial because of that Robertsonian translocation. All right, so looking at the ones that were viable, I'm going to make a little note in the top corner right here. That says of the viable ones. You'll notice that 1/3 are completely normal. 1/3 are carriers. And 1/3 have familial Down syndrome, which I'm going to abbreviate FDS. And you could also write this another way by saying that 2/3 have no Down syndrome, since both normal and carriers would not have Down syndrome. Okay, and then the last thing that I want to do really quick is chromosome count. So in our bottom zygote, this one right here, how many chromosomes would they have? Well, you should say that they would have 46 chromosomes. They have 2 copies of everything, and they're in their own individual spot. And the next one, they still have 2 copies of everything, but you have 2 that are attached. And if you were to look at the karyotype, these would have 45 chromosomes. And then in familial Down syndrome, you'll notice there's 2 at each spot. These would again have 46 chromosomes. So if we just were to look at a karyotype and see 46 chromosomes, we wouldn't necessarily know if that individual had Down syndrome or not, because both the normal phenotype and the familial Down syndrome have 46 chromosomes. All right, so that is the end of our Down syndrome section. Make sure that you can draw this out and you understand how this all works. And yeah, I hope all of that made sense.
I hope you found this video really helpful. The concepts and information presented in these videos will be true no matter what genetics class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Remember, if you are currently enrolled as a Baylor student, we offer free tutoring services. Our tutoring center is located on the first floor of the Sid Richardson building. You will find all the details you need to know about these services on our website, www.baylor.edu/tutoring. You may schedule a free 30-minute one-on-one tutoring session online through Navigate, or just drop in during our open business hours. For more information about our current services, please visit our website. Thank you.